Hello, my name is Karla Paniagua, and I am a PhD candidate of the University of Texas at San Antonio. Today, I will be presenting a deep learning algorithm for high-resolution spatial transcriptomic cell type identification by integrating gene expression and spatial information. Single-cell and spatial transcriptomics technologies are right now positioning as tools for the discovery of novel insights into the disease. Spatial transcriptomics characterizes tissue organization and gene expression at single-cell or subcellular resolution, providing a better understanding of biological processes. So this means that we get the information of the transcripts expressed in a slice of tissue and where they are spatially expressed. Cell typing is one of the key challenges in the single-cell and spatial transcriptomics data analysis, since right now there is no standard practice, so the use of different algorithms and cell markers results in inconsistent cell type assignment. This means that there is still a lack of reference or ground truth for cell type annotations. Another thing is that most of the current methods do not consider spatial information, where in some cases a specific cell type can reside together or next to other types of cells. For example, in here we see how epithelial cells in green are usually located together and close to endothelial cells in orange. To address these challenges, we use a deep learning algorithm which is powerful in capturing complex features even in high-dimensional data such as single-cell and spatial transcriptomics. In this project, we apply the state-of-the-art deep learning algorithm called Transformer, which has a multi-head self-attention mechanism that learns dependencies on the data and encodes contextual information, making it very popular and applicable to several areas, including single-cell and spatial transcriptomics. So this algorithm, what it's going to do is going to learn the context between the gene expression and the neighborhood of the cells. So with basically the spatial information of our cells. Our main objective is going to be to apply a deep learning algorithm based on transformers to investigate the inclusion of the spatial information of two targets. The first target is going to be the improvement of cell type identification based on the gene expression and spatial information. And our second target is going to be to study now the characteristics and spatial distribution of the different cell types and their transcription. So I'm going to go quickly through the pipeline, but the first thing that I need to talk about is that we're using a NanoString, which is a company data set, which is called, it's a high resolution spatial transcriptomics data set from non-small cell lung cancer. So right now, this is the highest resolution that we have in the market. The first step is going to be to do the cell culture, where we're going to extract the tissue and then the imaging. Then we're going to do the cell segmentation, where we're going to assign the transcripts to the different cells. Then we have to do the processing to identify the low quality cells and remove them from our data set, and just to keep the high quality cells. Then the dimensionality reduction, where we're actually going to do the clustering. And after this, we're asking, yes, but what we can use as a ground truth? Like, what can we use as ground truth? Well, as we talked before, there's no actual ground truth. So what we can use, or what we design to use, is the combination of two algorithms. So we're going to do this. We're going to do the cell type prediction on the cluster-based prediction, which is actually assigning one cell type to each of the clusters. And then we're going to do obtain the cell type prediction, which was done by a statistical method, which assigns a cell type to each of the cells according to their profile, and to like comparing to a reference which one it's more likely to be, which cell type is more likely to be, which was actually designed by the NanoString people. So this cell typing they already have. So for doing our ground truth, what we're going to do is to compare and to overlap these two different cell typing and obtain the cells that actually overlap between both of them. And with this, we're going to use those cells to train our transformer algorithm, which is going to end up in the cell type identification. So as a result, the first thing that we have is in this image, we have the cell type identification for NanoString company. In this image, we have the cluster-based cell type identification, and here we can see the overlap. So how many cell types overlap between the two cell typing methods? We see that 10 different cell types overlap, which is around 60% of the cells. Then where are those cell types under under transcripts spatially located? So we can see in this slide, we can see this is a slide of tissue, and we can see in different colors the different cell types. For example, in blue, we can see the tumor cells that would actually cluster together. And in this other slide, we can see the different genes expressed in the cells. So you can see in here, each of the dots, it's a gene expressed or a transcript expressed in one cell. Then, how accurately can our algorithm predict the cell types from the gene expression? So our model can actually predict the cell types with a very good accuracy using the gene expression, which is 96.3%. And we can see that it's very good in most of the cases. Then, do we see any differences in the spatial characteristics of the different cell types? And the answer is yes. We see that the tumor cells are smaller than the rest of the structural cells. Then, what cell types are in proximity with the tumor regions? So we found 6 different tumor regions, and we studied the cell types that were in proximity with these tumor regions to see which of the cells we have in more quantity. So in most of the cases, we can see that fibroblast was the cell with the higher proportion around the tumor tissue, except on tumor 5, when we can see macrophage is higher. This is the region tumor 5. So, as a discussion, our model can accurately predict the cell types from the gene expression. Spatial characteristics show that cancer cells are smaller than the rest of the cells. So we will continue studying the cells and their inter and intracellular features. And finally, we'll keep also investigating the inclusion of the spatial information. And in this case, what we're working on right now is including the transcript's spatial information. Thank you so much for your time.